now.
Fantastic. So people here already know each other. We just introduced Aitana. So that's great. Thank you for joining me. So I was thinking of making this somewhat conversational for you to see what's happening in our lab. And if there's any questions, if you wanted to tell me about what's happening to you, that would work as well. Actually, I've been enjoying that as well. So I'm going to switch the sharing. So I hope this is does this appear to be normal to you on your screen?
Yep.
Fantastic. So this is I listed 3 of ongoing things in the lab. The first is reviewing protein-protein interactions at genomic scale. So we are developing a technology that will change how protein-protein interactions are mapped experimentally and computationally. The second project I can briefly mention would be a spatial transcriptomics with ultra-high spatial resolution that would be able to review gene expression from human tissue in situ and retain the spatial resolution of where each gene is transcribed. And thirdly, listed a long name. This is for extracellular RNA sequencing technology that we've been developing and push that toward disease biomarker identification. We have some recent successes in applying this type of technology to develop blood-based tests for breast cancer recurrence and for Alzheimer's. So we have interest in expanding on this direction as well. So those are 3 of things I'm thinking of sharing with you. So the first is revealing protein-protein interactions at genomic scale. So current methods for reviewing protein-protein interactions include, for example, yeast-2 hybrid. And Clara here works on a number of things very related. Fred is one of them. There could be pull-down, mass spec is one of them. Purification-based and mass spec would be or co-IP. So these are typical methods. So if we worked with these methods, we know that these methods have different advantages. Nevertheless, if we wanted to review many protein-protein interactions from human tissue or from human cells, it's difficult for these methods to scale up. yeast-2 hybrid is perhaps one of the easiest to scale up. Still, it takes a center grant and many years of a center to yield on the genome scale of protein-protein interactions from human genome. So our goal here is to develop a technology that would enable a typical lab, say a typical bioengineering lab, that would map protein-protein interactions from human cells in a few weeks. And on the scale of the whole protein to whole protein interaction, which protein interaction with which, and suddenly gain explosive insights in novel protein-protein interactions relevant to the cell type. So this was actually the first version of this is under review. This was the PhD thesis from Kara Johnson. I don't know if she had interactions with you. She was actually quite active in bioengineering outreach activities as well when she was in the lab. She defended her thesis this summer. She's still in San Diego area. And the computational work is by another bioengineering PhD student, Zhijie Qi, they jointly worked out the first version that we are revision for publication. So the project here I'll be describing is going to be the next version of this, which will be actually simpler and will be directly applicable to live cells, large-scale protein-protein interaction. So Kara's design was that if the previously working with one protein at a time or many proteins at a time, working with proteins has its challenge. So Kara was thinking if we can tag every protein with a unique DNA barcode, and then if we can harvest all the interacting protein pairs and sequence their barcodes, so then we would be able to leverage the very high throughput DNA sequencing technology to sequence out protein-protein interactions. So that is the overriding theme. So hello, Lex. Thank you for joining. So when we're talking about the technology of sequencing protein-protein interactions, revealing protein-protein interaction by sequencing. So when Kara had an approach that would ligate or conjugate a unique DNA barcode to every protein, or nearly every protein, and then she can harvest the proteins in interaction and harvest their companion DNA barcodes. And by paired end sequencing the barcode pairs, essentially she sequenced out protein-protein interaction pairs. So on the panel E, this is a partial map of the reviewed protein-protein interactions specifically related to splicing complexes, and actually that correlate to our knowledge quite well and already have a number of interesting new protein interaction leads. And these can even be updated to interrogate protein RNA interactions using a similar strategy where one protein is tagged with DNA barcode, the other end would be the entire RNA library. So the ongoing project in the lab and also can serve as a rotation project if someone has good, very good strong interest in molecular aspects, would be make a version 2 of this that would enable the conjugation of protein and DNA barcode in vivo in live cells. So that we have a design strategy of doing that. We are testing that. So the other second ongoing project recently, in the recent 2 years, actually, this has been funded by both the NIH Common Fund HubMap, the Human Molecular Atlas, NIH project, and Chan Zuckerberg's Human Cell Atlas project as well. So the goal, so the title is Spatial Transcriptomics with ultra-high spatial resolution. So the problem is that although in recent 3 to 4 years, people have been able to assay gene expression with spatial information, namely that if I were to look at, take an example, if I were to look at panel C here, in the lower part of panel C, what we are seeing is a piece of the human vasculature. So in panel A, this is a piece of human artery from a diseased donor. If we were to section that vertically, then we would have a ring that is a section of the human artery. If we were to cut the ring into 4 quadrants, we are seeing in panel B, A, B, C, and D. Those are 4 quadrants of a circle of a section of the human artery. And in C, we are looking at one of them. So colored here are fluorescent labels of cDNA that are reverse transcribed and generated from the RNA or messenger RNA of this piece of tissue. Then if we were to utilize so-called spatial transcriptomics technology, we can derive and sequence out the transcribed RNA at the grid on this spatial slide. So between the grids, currently the resolution is approximately 10 microns. This is probably on the size of a large, very large cell or several cells. So at every grid, we can add unique sequencing barcodes. So therefore, after RNA sequencing, we can obtain the information of at every spatial resolution at 10 micron resolution, what are the genes transcribed there. So essentially, we have a spatially resolved transcriptome of human tissue section. So this is a status quo, a very recent status quo. We recognize that the 10 micron resolution may or not be perfect. If people are interested in delicate tissues, including the brain, we are very much interested in the human vasculature. For example, the endothelial cells, the endothelium, the very inside layer of the human vasculature is a single layer of endothelial cells. These cells, on average, would have a 1 to 2 micron of diameter. So there, this 10 micron resolution currently would not enable us to precisely see what is in the insidemost layer expression level of the human vasculature. So we'll be mixing some of the other fibroblast, smooth muscle. We are not going to be able to completely dissect every piece of resolution. So our deliverable is to develop a version of this, but improve the spatial resolution to sub-micron level. And we will also have a lot of other advantages, include the dynamic range and so forth. So what Tri, a postdoc in the lab, leads, and Xue Sherry, she's a BSMS student working with Tri. What they have designed is that they actually reutilize a previously used Illumina flow cell. After sequencing, these flow cells usually were discarded previously. But Tri recognized that the Illumina flow cell is actually a very finely printed barcode microarray. So each small nanowell in a printed Illumina flow cell has an average distance between at approximately 0.1 to 0.2 microns using the latest version. So in fact, after a usual Illumina sequencing, when the flow cell was thrown away, it always thrown away, it's a barcoded microarray with extremely high resolution. If we ask even the best manufacturer facility in the world, there are not many. There are probably just 1 or 2 in the world that can really do this very fine printed DNA barcode microarray. We Tri figured out a way to essentially recycle those. And now that we have this, instead of the current 10x genomics microarray that has a 10 micron or worse resolution with only a few thousand probe sets on the spatial array, now we have a microarray free of charge that is more than 100-fold better in resolution and more than 10,000-fold more in number of grids to resolve. So this has now been a technology transformative project funded by NIH HubMap, and Tri and Xue are testing this technology on human tissue. So we are working with people on campus with lung tissue, with vasculature tissue, and with heart tissue, and to try it out. So are there questions so far for the first 2 possible things? Okay, if not, I'll be briefly going to another major endeavor here in the lab. So this is about development of blood-based diagnosis for human disease. So most likely that UCSD will initiate a new blood liquid biopsy disease biomarker center that I will be leading that center. This is a core technology will be used here is what I'll be describing here. And so this, I call this project categorization of blood serum extracellular RNA by sequencing and development of disease biomarkers from liquid biopsy. So first of all, I would like to say a few words about extracellular RNA. So these are RNA, but these are RNA not inside the cells. We usually understand that RNA are transcribed and work inside the cells. Those are the majority of RNA. I would say out of 1,000 RNA molecules, 999 of them are inside the cell. There are very few RNA that are actually secreted to the extracellular space. These are called extracellular RNA. So since these extracellular RNAs were discovered from human blood since the year 2000, actually this has gained now very recent huge momentum as media for detecting human disease because they assume they are secreted. Then if they can get mixed in the human blood, then you can potentially see them, detect them from periphery blood. And suddenly, if this RNA were secreted from any tissue of interest to you, including the brain, if you can detect it in the periphery blood, this will be a convenient tool. So these RNAs do not completely dissolve or get digested by RNAs because they often get protected by some extracellular carriers. Their concentration are very low compared to the typical cellular RNA that we are working with. So this is probably 1,000-fold lower in concentration than the typical RNA we know about. And the sizes tend to be small. So from 20 to 150 is based on our own data. So you can see that this includes small RNA, but also fragments of long RNA. If we are more familiar with messenger RNA can easily have an average size of, say, 1,000 bases. Now here, the longest extracellular RNA that we see from the human blood are only 150 base-ish. So that means that there's no full-length messenger RNA there. There could be fragments of the messenger RNA that can be detected. So it turned out that when people wanted to develop RNA-based biomarkers for human disease, if people wanted to do that, hello, Ming. Thank you for calling in from the East Coast.
Hi, sorry for being late. I just saw the email.
Another problem. So we meant this to be a casual conversation. Most people audience here are UCSD graduate, PhD, bioengineering PhDs, most of them. But thank you for calling in. And so this is more of a casual conversation of what's happening in my lab. And if people have any interest in taking any of this as participating in a rotation project, that will be basically a casual discussion.
That would be great. Yeah. Really looking forward.
So since you got in, we were talking about the third possible project, actually ongoing project. This will be expanded to actually a UCSD center of liquid biopsy diagnosis. But the gist of this is to have a technology to detect extracellular, the secreted RNA from human blood and see if those detection can be used to develop disease biomarkers. So that is the crust of this. So there are RNA that are secreted to outside of the cells. The concentration of this extracellular RNA are much lower than cellular RNA. And with your background, indeed, yes, some of the immune cells actually dump extracellular RNA as well. And we actually have been applying for a phase 3 clinical trial for some of the autoimmune disease based on extracellular RNA of the human blood. So the challenge of developing any disease biomarker is in its clinical validation. If we are developing a blood-based biomarker, if we wanted to test in a clinical cohort, so in some of the examples later, I will show a cohort. We are following up breast cancer patients after chemotherapy to see if there are extracellular RNA that are indicative to recurrence of breast cancer. You need to follow them for at least 5 years, right? Because there are very few recurrence within the first couple of years. Unfortunately, there are, but very few. To develop anything, you at least have to follow them for 5 years at the minimum. In another example, I'll briefly show is to have a predictive biomarker for late onset Alzheimer's disease. So these are people who are developing Alzheimer's in their 70s. Those are elderly people. Most 95% of Alzheimer's are this late onset. They don't have the most critical mutation of Alzheimer's. If people carry those mutations will unfortunately develop Alzheimer's much earlier, but those were only a small percentage of the Alzheimer's patients. So those people, in order to develop any biomarker that's predictive, you really have to test by follow up with normal cognitive elderlies, follow up sufficient years to see if your detected molecular change predates the clinical diagnosis of their Alzheimer's outcome. In the example I'll be showing here is a 15-year follow-up study. So the thing is that if we have to develop some biomarker based on blood sample that would require typically for DNA sequencing, RNA sequencing would require a few microliters. I'm sorry, a few milliliters of blood. That's typical. You will have to really draw the blood from patients. Then these validation studies will take 5 to 15 years. So that is a bit challenging. That is a challenge in the field. So we started to see actually from clinicians and blood banks that if you have a technology that can work with only several microliter of blood, for reference, a drop of liquid on average is 30 microliter. That is a droplet of liquid. If you only use a partial droplet, say 10 microliters, so that is something that ongoing trials can spare, right? If they give you 10 microliters of their previously drawn blood, there's nothing. It's in the error zone of the nurse. So the ongoing trials don't care do not feel huge sacrifice if you give you 10 microliters of blood for developing a new test. So knowing that, suddenly we understand that if your technology can be based on a tiny amount of liquid biopsy, suddenly you can change the landscape of validation, of clinical validation, because you can suddenly jump into ongoing trials, because typically they would draw blood, but then they can have a sufficient amount to add the metrics that you would like to measure. So this is what inspired us to develop this technology. So this technology is called a silverseq. This was published a couple years ago from Josh Zhou as a previous currently about engineering alumni. I would like to disclose that Josh actually works in a startup company called Genomic. I'm a founder of that startup as well. Okay. So silverseq is an acronym for Small Input Liquid Volume Extracellular RNA Sequencing. What this technique works on literally a droplet of liquid biopsy, say human blood serum, human blood plasma, and it can carry out the RNA sequencing of whatever RNA in that small amount. When we were thinking about this technology, we asked ourselves, was it possible at all, right? It started with such little material. We thought it was possible because a drop of blood times the concentration of extracellular RNA would equal the amount of RNA in that drop. That amount is roughly on the same scale of the amount of RNA in a single cell. So we now know that single cell RNA sequencing, since its invention in 2009, now it's 11 years, it has become a popular technology. And so people now accept that we can probably reliably sequence RNA from a single cell. Now, knowing that the amount of RNA from a single cell is about the same amount of RNA molecules from a drop of liquid biopsy. So we don't feel there is a physical barrier for not able to do that, but actually working it out, because these RNAs are more fragmented. It has its own characteristics. Turn out that it still took a couple of years to work out this technology. Just to recap, so the status quo is to do sequencing either RNA, DNA from typically a milliliter of liquid biopsy. That is about this tube is a 2 milliliter tube. And whereas silverseq can reliably work on 5 microliter of human blood serum. So this is about 1/6 of a drop of blood. So if I were to very briefly show 2 examples of how this was applied. So in one example, I will directly go to the final outcome. So we were testing with Dr. Irene Su here at UCSD Morris Cancer Center. She has run a clinical.gov registered trial for a woman who have undergone chemotherapy for their breast cancer. And she were able to, in her 5-year follow-up, harvest a number of serum samples from the woman who have unfortunately had a recurrence of breast cancer after chemotherapy. And she also, within the same follow-up cohort, she also had collections of serum samples from people who have not exhibited any recurrence. Those are fortunate. And then we, after silverseq-based sequencing, so we ask ourselves, can we develop any predictive biomarkers? And we actually wanted to make it more challenging. Can we use any of the commercially available gene panels? Those are gene panels released from with public knowledge. So these are not any specific gene that we find. If we find any gene feature, it's actually easier for us to make a great test. We are using publicly available gene sets and using them. So this is a ROC curve. This curve reflects how good a test is by changing the threshold. Typically, if this curve is shooting up and going flat with a lot of area under the curve, it is a great test. So if the ROC curve is more of a diagonal, this is not a good test. And in this example, through cross-validation, we are showing 3 cross-validation results here. We've done 100 of them. And so these ROC curves are going quite well. The area on the curve is big, meaning that they seem to be very reliable. And this even doesn't rely on genes that we find. We are just using previously released gene sets. Those gene sets were not even developed from extracellular RNA. They are typically developed from tissue biopsy, and they were used for grading the cancer or predicting metastasis and with different utility. But here it looks like that even the publicly known gene sets plug in extracellular RNA sequencing. We now seem to have a good indicator of cancer recurrence after chemotherapy. There's a question from chat. Thank you, Clara. Yes. Okay. And so if I were quickly to divert into another published application, so this is the goal was to see if there's any blood-based biomarker we can develop for predicting Alzheimer's onset. So this was just a little bit of background. There are at least 2 types of Alzheimer's, the early onset and late onset. Early onset are people who carry mutations in a few genes. Typically, there are 4 genes that we know if there are mutations, people have increased chances of developing Alzheimer's. These people typically would develop Alzheimer outcome before they become elderly. But this is only a small portion, even within the Alzheimer patients. 95% of the Alzheimer patients do not carry a critical mutation. So these people develop Alzheimer's typically at a later stage. So here we only worked with people of 70 years old or older. So when they get enrolled, they were already 70 years old or older, and they were being followed up for 15 years. So here, still currently we do not yet have any outstanding predictive biomarker for Alzheimer diagnosis. If we were to have this, perhaps will change how clinical care is provided or treatment is provided to Alzheimer's. So among these people, the first 9 people were diagnosed as normal in their cognition, and throughout the 15 year of follow-up, they were always normal. So there's no problem with them. So this became our controls after the 15-year follow-up. So there are AD group, there are 15 of them. When they came to the clinic, they were already diagnosed on the first visit, unfortunately, into Alzheimer's. And after their deaths, the autopsy also verified AD pathology. So these people are the Alzheimer group. So they were also followed up for 15 years. There's a third group from this cohort where when the elderlies came into the clinic, they were diagnosed as normal. But sometime during this 15-year follow-up at the time of a Red Cross, at that time, they were clinically diagnosed into mild cognitive impairment, or Alzheimer's. Those people who were diagnosed as cognitive impairment later also diagnosed into Alzheimer's, and their autopsy confirmed that these were Alzheimer's disease. So we have these three groups. It turned out that now, at this point, in the 15 years, their blood sample were drawn. But as you can imagine, there have been many studies carried out on these blood samples already. Now there are not much left. And this was one of the things that when we only need a few microliters of serum, it actually worked out for us. We were still able to assay the remaining sample here. So I wouldn't go to the detail of the statistics, but it turned out that there is one gene whose extracellular RNA exhibited a strong upward trend in the time course of those converters. Turned out that so for the controls and the disease group, so these extracellular RNA throughout this 15-year follow-up are more or less flat. So this is a regression coefficient showing how steep is the fitted curve for those controls. You can see that for those controls, they are mostly flat. Whereas for converters, so this fitted curve always shoot up. So for all the converters in this cohort, this extracellular RNA increase with time. So this now become a biomarker for a predictive indication of disease Alzheimer outcome. So with that, I wanted to summarize. So we now have a technique for sequencing extracellular RNA from a single drop of blood serum or blood plasma. So this is called a silverseq. And for cancer versus non-cancer, cancer recurrence versus non-recurrence cases, it looks like that through sequencing the extracellular RNA, we are able to develop a reasonable classification of cancer recurrence. So this, we are pushing it forward for the new center we are building, a liquid biopsy center that we are building to develop into a means of predicting recurrence. And hopefully this can change how these patients are monitored and are treated for their possible recurrence. And also, we seem to have identified extracellular biomarker that is pre-symptomatic. These people have not developed cognitive symptoms yet, but it can seem to be able to predict their symptomatic diagnosis. And so we would like to test this with much larger cohorts now that we are lining up with several cohorts from the nation to test this further. As for a rotation project, we would like to dive into the mechanism a little further. So the background is that there's little known about how the extracellular RNA can dodge extracellular RNAs and remain in human blood. As you know that the human blood has plenty of extracellular RNAs. They are there for a reason, because for most of the virus who come into the bloodstream, these RNAs just cut them. So that's one of our protective system of mammals. So for this reason, for a long time, for many decades, people do not even think that there are extracellular RNA. But this was, so since year 2000-ish, extracellular RNA was discovered. And now that people can actually use that for diagnosis purpose, there are prenatal tests, for example, that are based of extracellular RNA from the fetus in the mom's blood. The project goal here is to really dissect how can extracellular RNA exist or retain in the human blood without being digested completely. And knowing this mechanism can help us to refine the detection technologies and also just appreciate whether these RNAs are there for a reason. They may serve as a signal molecule. So the project goal here is based on our preliminary data that nucleosomes, these are a group of proteins that can protect extracellular RNA. So the impact would be that if this is tested, verified, So this will reveal a new mechanism of extracellular RNA protection. So this will solidify further development of extracellular RNA-based biomarkers, or understand how extracellular RNA may be a communication or signaling molecule from cell to cell, from tissue to tissue. So I feel that if we were to do it as a rotation project, this would be if we can IP immunoprecipitate histone proteins and sequence the co-precipitated, co-pulled down RNA to see what these RNA species are. If first of all, if there are any of them, and secondly, what are those? So these would serve as the initial test data for testing whether these nucleosomes or histone proteins are a carrier of extracellular RNA. We have carried out some initial experiments, and we have already have initial data along this line. So with that, I did a rapid fire shot through of 3 things happening in our lab. This year, our lab is funded by several national centers. We are a couple of national centers, and we are becoming a new UCSD liquid biopsy center. So this is only a subset of ongoing things, and we certainly can have great interest of expanding on several things along these lines and beyond these lines.
Hi, Professor.
Yes.
Oh,
Carl.
thanks. No, this is really interesting. And this last project that you're talking about is something that I've actually looked a little bit into. So being in the Metallo lab, we've looked at PhDDH and both sort of the activity and the need of serine for production of glycosphingolipids, sphingolipids, and a lot of other signaling molecules, but also just localization molecules for proteins. So I'm just curious, here you're seeing an increase of PhDDH in the bloodstream, or the mRNA for it. Can you localize where it's coming from, either by looking at the nucleosome that it's bound to, or is there some way to sort of localize what, where this extra expression is coming from?
Indeed. So that we have went back to several AD tissue banks and published data from several AD tissue banks. So the just background information for everyone. So this particular gene PhDDH actually is a required enzyme for metabolizing glucose into serine and glycine and serine. So glycine and serine are, first of all, required nutrients. And secondly, they actually help on synapse. Hello, Imran. Thank you for joining us. So there is a huge interest in general in glucose metabolism, serine synthesis, glycine synthesis role in cancer metastases and in Alzheimer's, and more recently in Alzheimer's. So Carl, you are an expert here. So PhDDH is mostly expressed in astrocytes in the brain, and it's required for brain development. If PhDDH is mutated in humans, humans will not develop into a full-grown brain. It will grow into microcephaly. So it's a small brain, unfortunately. So because of lack of this required nutrient and perhaps many other problems associated with it. So then we leverage in the brain, human brain data. So we can indeed narrow down to increase the PhDDH, actually surprisingly, in microglial, in the brain, gliodendrocyte precursors in the brain. And we have recent data from mouse AD models in collaboration with Xu Chen's lab here in neuroscience at UCSD, that we started to see specifically in hippocampus AD mouse model, but there are many AD mouse models, several at least in the model, AD model that Xu's lab works on, mostly for tau pathology. We specifically see increased PhDDH in the hippocampal area, and we are collaborating with several labs in different universities to test on different mouse models. Having that said, there are indeed people reporting possible non-change of PhDDH in the brain. And there are also results showing that if you supply D-serum to some AD mouse model, you may have cognitive compensation. So these argue for a reverse trend of which way should they go. We certainly have certain conjectures here, where depending on the course of the pathology at early points, if you superactive DNA receptors, there may be some cytotoxicity leading to early onset. And at a late stage, now you don't have a sufficient serum supply. Many things go wrong, synapses go wrong. At this point, if you then resupply the serum, perhaps there could be a rescue effect. So Chris and I actually had some brief conversations several times related to this. So I appreciate that there is a lot of complexity going on, and I certainly would love to get more insights from Carl and Chris from your lab on these lines. In a nutshell, we are narrowing down more in the hippocampal region in AD. Nevertheless, I'm not saying that I'm saying it's a time course. I understand that the direction of change can depend on the pathology time course. And it doesn't mean that it has to be always high or always low. And certainly this may perhaps makes it more interesting. And the genetics data showing that the other way around, the lacking of PhDDH has certainly a number of developmental problems. And we can imagine that in adults, if you severely depleted of this required nutrient, there would be brain problems as well. Carl, does that start to get back to your question?
Yeah, it's just interesting. What you're saying, the diversity of sort of the uses and the necessity for serine makes it more complicated, especially in the brain where now you have D-serine also as a different signaling molecule. It's not just yeah.
Yes, our working hypothesis is that at early stages, too much glycine and serine produced too high increase. We indeed see increased PhDDH at least in the relatively early stages of AD models, and we feel that this may be related to excitotoxicity. This would in line with overactivation of the NMDA receptor leading to excitotoxicity, which is a more of an established pathway of leading to AD etiology. But later, then you probably need to resupply the serum because at that point, many of the neurons already have problems. Now synapses become lacking, and you need to reestablish synapse. At that point, resupplying the serum perhaps would be a good thing. At a later point, perhaps activating PhDDH would be a good thing. So this is our working hypothesis. So there's a question from Phoebe. Oh, okay. Yes, thank you, Phoebe. Yep. And with Carl, I didn't mention that we also have a recent publication with Liangfang Zhang's lab related to cell surface molecules. Specifically, we seem to discover cell surface RNA. Then that we would have a lot of interest in understanding how that interact with lipids as a way of stabilizing RNA.
Hi, Professor. Thank you for the cool talk about your research. I do have a question that's a little less related, but I think you mentioned that you started your own startup company. I wanted to just ask, like, what's it like? What does it take to start your own
That's a
company?
wonderful question. So may I get back to you separately, or if we have some interest group on that, because that could take time, and then the best way is to interact with you to see what's going on with you, and because it's very different, and I only have a tiny piece of experience, and certainly we have many faculty members here in Jacobs School of Engineering have a lot of interest. Yeah. So may I get back to you more individually or separately as small group?
Okay, thank you.
Well, thank you for joining me, and also thank people for calling in from different parts of the world. Imrat calling from India, Aitana calling from Argentina. And thank you, everyone. And Ming calling from Boston. Huh? Thank you, Carl, Josh, for sharing this.
Yeah, no problem.
Thank you for the talk. It's really cool stuff, and it's sort of a very interesting and growing field.
Thank you, Carl. I look forward to learn more about your work.
Honestly, me too.
And Lex, thank you for calling in. Would you like to introduce yourself to us? Okay. Okay, thank you, everyone. Take care.
Thank you.
Thank you.